Can everyone see the slide?
हाँ सर.
हो सर.
So yesterday we studied this microbial cultures, how they are important, and how the collection center works out. So next is enzymes. Now you know the importance of enzymes from detergent powder till everywhere, everything, every living reaction is carried out by enzymes. So industrially, this lot of enzymes have been isolated and they're used. So what are the sources of enzymes? First important source is microorganisms. In molecular biology, in PCR, which enzyme we use? कुठला एन्झाईम आपण वापरतो in PCR?
टॅग polymerase.
Tag polymerase. So isolated from bacteria called Thermus aquaticus. So similarly, we have got restriction enzymes are there. Then amylases are there, proteolases, proteolytic enzymes are there, lipases are there. All these etc. enzymes we are using. Second source is animals. From animals we extract lipase, trypsin, renates, etc. Then we have got plants. From plants, one of the important enzyme isolated is papain. From where papain is isolated, can anyone tell? पॅपेन कशात आयसोलेट करतात? कॅन एनीवन टेल? हॅलो, आवाज सगळ्यांना ऐकू येतोय?
हो सर.
Then from where it is isolated papain? सांगू शकता कुणी? Everyone has had papaya? पपया फ्रूट खाल्ले का सगळ्यांनी? खाल्ले का पपया?
हाँ सर.
So papain is isolated from papaya fruit, raw papaya fruit. Raw means there is ripened papaya which is yellow or red in color, and raw is green color. So after harvesting this papaya fruit, what they do? They rupture the papaya fruit with some instrument or knife, and whatever the white color fluid flows through this papaya fruit, आपण त्याला चीक म्हणतो. So they harvest this sap and they isolate the papain from this sap. Then remaining raw papaya is there. After the damage, it will not ripen for selling it as a fruit. What they do? They will convert this into चिलीमिली we call it. जे चेरी असते पपया चेरी त्याच्यामध्ये कन्वर्ट करणार दिस रॉ फ्रूटला, अँड दिस इज द बाय प्रोडक्ट, अँड मेन प्रोडक्ट इज दिस पॅपेन. सो दिस पॅपेन इज what it is a protease. So where proteases are used? These amylases proteases, they are used in where? In medicine, where they are used for digestion. The medicine which are given for digestion, if anyone has got acidity, all those things, what they will do artificially, they will provide enzymes. These enzymes will degrade the undigested food. So papain is isolated from papaya fruit. सगळ्यांना कळालं? Then applications of enzymes, wine making is there, cheese making is there, production of alcohol आहे, treatment of wastewater, metal mining आहे. In metal mining, how they do it is bioleaching. For example, if here we are harvesting these minerals for around 200 years. Now what happens? There are rich sources of mineral. म्हणजे फॉर एक्झाम्पल एफीचे सोर्स आहे, आपण फेरस साठी आपण काय करतो, मिनरल डिपॉझिट घेतोय. So some deposits they will have ferrous content above 70 or 80%. So it is easy to use chemical process. But some rocks they will have ferrous content below 20-30%. So there the isolation is very unprofitable. We can isolate, but the profit is very less. So there we can use this microorganisms. But ferrous is not a very what we call expensive metal. So expensive metal like silver, gold, if the quantity is less, but it is hard to extract by chemical method, then we can use biological methods, bioleaching we called it. So what happens? This bacteria we culture on that ore, and the bacteria will secrete some enzymes, and this enzyme will leach out the gold, silver, all these precious metals. So there also these enzymes are used. Then measurement of glucose and other constituent in the body fluid. ग्लुकोजच्या मेजरमेंटसाठी पण एन्झाईम वापरले जातात. And also to cover the genetic disorders caused by enzyme replacement. So here these enzymes are used. So a lot of applications are there for enzymes. So how bioinformatics can be used for enzymes? I've given you example last time of DNA polymerase. So we have took this DNA polymerase from Thermus aquaticus. Why? Because it is stable at above 95 degrees Celsius. But human DNA polymerase is not, tag DNA polymerase is not stable at such a high temperature. Function एकच आहे दोघांचा, add on nucleotides and synthesize the new strand. But what happens? Stability matters, and stability it is based on which amino acid constituents are present in that enzyme or peptide. So you can study what you'll do. You'll isolate Thermus aquaticus, tag DNA polymerase gene. Similarly, what you'll do, you will construct this forward and reverse primer, and you will isolate it from other organisms this gene. After that, you will get amplification. You will go for sequencing. From the sequence ATGC, you will get amino acid sequence. mRNA from mRNA you will get amino acid sequence, and by looking the amino acid sequence you can know which amino acids are responsible for stability of these enzymes. So here bioinformatics is used. It is one of the application. Then comes drug design. Now drug design, it has got how many branches? See, first is bioinformatics, second is protein modeling, structure based drug design, then chemoinformatics, rational drug design, and pharmacophore based drug design. So chemoinformatics, what they do, similar like bioinformatics, what do they do? Chemical reactions they can put in soft lab. We do chemical reaction in what acid base reaction is that we'll do in a beaker, burette, pipette we'll use. But here they will use nothing, but they will develop programs, and in these programs they will run the reaction. So last time I explained for discovery of new drug, it is very lengthy process, and if you are registering for patent, you have to discover and find its application more rapidly and launch that product in the market. Then only that company can earn the profit. Next come protein structure. Now protein, it has got primary structure, secondary structure, quaternary structure, and tertiary structure. So all these structures we can artificially look. We need not to, if you want to look for structure of protein, what you have to do? Secondary structure or quaternary structure, you have to isolate that protein in a pure form. First, you have to grow the cell. For example, if you are, for example, take you are studying cry protein, what you will do? You will cultivate this Bt cotton, Bacillus thuringiensis in a liquid media. Then what you'll do? You will harvest that bacillus. Then if the protein is extracellular, what you'll do? You will discard all the cells and take the supernatant. From that supernatant, you have got hundreds of proteins. You will isolate specifically the cry protein by ion exchange chromatography. Then using, then after separation, you will elute that cry protein. Then you will dissolve it in 100% pure water, and the protein will form a three-dimensional quaternary structure. And then you will study this by X-ray crystallography. Then you will get the data of this, and then you will study the 3D structure. So how much lengthy process is there? Here what you have to do, just by looking at DNA sequence, you will construct a primary structure of protein, and that you will put in a software, and you can watch 3D structure. In few minutes or few hours, you can study the 3D structure, active sites, and how it will react to pH, buffer. All this reaction you can develop in a soft lab. So these are applications of bioinformatics in protein structure. Now, classification of protein you have studied: simple, conjugated, and derived proteins. Importance of proteins, you know, all enzymes are consist major part of proteins. So structurally also, our cell membrane is present of peptidoglycan, and structurally our biomass contain protein. And they water our body make their maximum part is due to protein. Functionally all enzymes act as a, means their present maximum quantity is present is protein. So enzymes carry out all the reactions in the living organisms. Now this DNA replication, if you study DNA replication, DNA transcription, RNA translation, and protein translation. So this is a step by which protein is synthesized. Now chromatography. Everything, whatever the metabolites is there, if you are not able to isolate and find its correct activity, there is no use. Commercially you cannot sell, whether it is antibiotics or high purity chemicals. You have to separate it from the mixture, and chromatography is the basic principle which is used for separation of these molecules. So there are different types, that is partition chromatography, absorption chromatography. सगळं तुम्हाला केमिस्ट्रीमध्ये झालेलं आहे. म्हणजे पार्टिशन मध्ये यू हॅव पेपर अँड थिन लेअर, गॅस लिक्विड आहे, अँड ॲब्झॉर्प्शन मध्ये कॉलम, आयन एक्सचेंज, जेल एक्सट्रॅक्शन. दिस ॲब्झॉर्प्शन chromatography is used for isolation of enzymes and antigen, antibodies, vaccines. This is used. Then how this biotechnology interdisciplinary field? So we are using engineering, genetics, biochemistry, molecular biology to study basic life science disciplines, and the applications they are used in biotechnology. So biotechnology is nothing but interdisciplinary field. Now bioengineering, biotechnological processes. So how the product is formed? First what we'll do, they will do upstream processing. So this, for example, we are taking example of, say, which insulin production, for example. So first they will do upstream processing. Upstream means to increase the biomass. What you will do in this step, you will prepare a liquid medium, autoclave it, you will add the required chemicals in that medium, ingredients. For example, you are producing Na broth, nutrient broth. Then you will add all the chemicals, salts, and this peptone and all those things, yeast extract. You will autoclave that, and you will add a inoculum in it. Then fermentation of the fermentation and biotransformation. So these microorganisms, you will provide exact pH, exact temperature, all the nutrients. So at optimum conditions, the microbial biomass will grow. It will constitute for 5 to 50% of the total fixed and operational cost. So major economic is here in fermentation and biotransformation. And these organisms, they will produce the metabolites. Then you go for downstream processing. Downstream processing means separation of the cells and fermentation broth. For example, the protein is intracellular, then you need cells. If the protein or your active product is extracellular, you require broth. And then you use here chromatography techniques, different chromatography techniques, and by that you can isolate the pure product. And then what happens, you get your pure product in a crystalline form. For example, you are getting this penicillin. So penicillin मिळालं तुम्हाला crystalline form मध्ये. So that penicillin, it is not made into easy capsules. First you have to add what, pillar material. So if you see this antibiotics and prescription, what is the medicine is there, if you see on their backside, they will give, it will contain 100 mg, 500 mg. दिलेलं असतं ना, active concentration. For example, paracetamol is there, you will study, you look 500 mg is there. So 500 mg is the quantity of that paracetamol, but the weight of that capsule is not 500 mg, it is 1 gram or 2 gram. So remaining is filler materials. So these filler materials are starch, कॉर्न स्टार्च आहे, देन टॅल्क आहे काही केसमध्ये. सो दिस टॅपिओका स्टार्च इज यूज, and in this filler metal this active component is mixed. They are converted into tablet or capsules, and they are used for further packaging. So this is the exact process by which these large scale antibiotics or active products are produced in microbial fermentation technology. ठीक है. So here we have covered the basics of your bioinformatics. I've discussed you how bioinformatics is important, how it has shaped the research in biotechnology. So next time, from next lecture, we'll study the computer used in bioinformatics. What is the role of computer or different technologies developed in the computer field which are used in bioinformatics, and we'll go on with this. Okay. Hello. Hello.